Hello everyone. Today let's learn something about helminths and anti-helminthic activity. I am Supriya and let's learn something about helminths and how do we tackle them. So, helminths, we are interested only on helminths that are parasitic because there are many other helminths which are not parasitic in nature, but we are concerned only about the parasitic helminths. So parasitic helminths can be divided into two parts, which is flatworms or platyhelminths, and the other one is roundworms or nematoda. So in flatworms or platyhelminths, there can be flukeworms or tapeworms. So these are the two divisions that can be divided. So let's talk about the mode of transmission. So transmission in helminths can be of different types. Some of the helminths can be transmitted by accidental ingestion of infective eggs or larvae, which include Ascaris, Echinococcus, Enterobius, Trichuris, and some hookworms. So such kind of helminths can be directly transmitted by the accidental ingestion of infective eggs or larvae. Again, some of them can be penetrated through skin, which include hookworms, schistosomes, strongyloids. So such kind of helminths can be penetrated through the skin. Again, there can be some intermediate host vector, which can be an arthropod vector of filarial worms. So these can be some mosquitoes, which causes filarial worms. So this is one of the transmission mode of transmission, and again they can be transmitted by consuming the intermediate host like clonorchis in fish, tapeworm in meat and fishes, trichinella in meat. So such kind of intermediate host when consumed directly can be transmitted. Some of the common soil-transmitted pathogenic helminths of the world. This includes roundworm, whipworm, hookworm, strongyloids. So these are some of the common. In roundworm, the most common is Ascaris lumbricoides. In whipworm, it is Trichuris trichuria. In hookworms, it can be Nicator americanus and Encyclostoma duodenale. So these three kind of pathogenic helminths, they can be diagnosed easily, but strongyloids stercoralis, this is the pathogenic helminths which require a very different diagnostic methods. This is not easily diagnosed, so it require a different type of diagnostic methods. Again, let's see the human helminth affecting globally. So these are the most common helminth infection prevailing in the world. You can see that Ascariasis is the most prevalent. So it has affected about 807 million people, and the least affecting helminths is dracunculiasis. So it has affected about 0.01 million people. So there are also other helminth infection like which is caused by trichuriasis, hookworm, strongyloidiasis, lymphatic filariasis, onchocerciasis, schistosomiasis, foodborne trematodiasis, cysticercosis, and loesis. So these are some of the commonly affecting human helminths in the world, and the most common is Ascariasis. So you can see the prevalence of soil transmitted Helminth diseases. This occurs mainly in children of the age group of 1 to 14 years. So it is all over the world, and about 24% of the children are globally affected, and in India about 68% of the children are affected. So apart from this, 28% of the children are at high risk globally to such infection. So you can see that about 2 billion people are infected globally. So in number it is 2 billion people globally, and about 870 million children, they live in such an area of high prevalence. And like in this is common in Africa, South Asia, South America. So these are the places which are most affected by such diseases, and India alone contributes nearly about 25% to the global cases. So we can say that this is mainly due to the unhygienic conditions of the children living lifestyle. So this can be the main factor for the prevalence of these diseases. So the current WHO guidelines is presented here. So according to the WHO guidelines, they in an untreated population. So in an untreated population, when the prevalence is less than 20%, so there is no need of preventive chemotherapy. But if the prevalence is between 20% to 50%, so then preventive chemotherapy is done once a year. And if the prevalence is more than 50%, then preventive chemotherapy is done twice a year. So in this treated population, it is evaluated after 5 to 6 years of preventive chemotherapy. And in this evaluation, when the prevalence is less than 1%, then there is no need of preventive chemotherapy. When the prevalence is between 1% to 10%, then chemotherapy is done once every 2 years. When the prevalence is between 10% to 20%, then the preventive chemotherapy is done once a year. And if it is between 20 to 50%, then chemotherapy is maintained like a previous frequency. And if the prevalence is more than 50%, then the chemotherapy is done every three times a year. So this is the scenario which was previously followed, but there are current WHO guidelines which are modified guidelines, and according to this, when there is prevalence of 1%, then there is no preventive chemotherapy. When there is prevalence of between more than 1% to 50%, then preventive chemotherapy is maintained like the previous frequency. And when there is prevalence of more than 50%, then the preventive chemotherapy is done 3 times a year. So this is how they have made a table for guidelines even of treating the population. So there are many allopathic drugs recommended for Helminth infection. So in market you can see such drugs like albendazole, mebendazole, parental palmate, levamisole. So these are the drugs available in the market. Out of this, WHO recommends albendazole 400 milligram and mebendazole 500 milligram. So apart from these, there are many other generic and brand names of these drugs, which include Albenza, Biltricide, and many more. as it is given here. So traditionally, there are so many medicinal plants that act as an anti-helminthic activity. So such kind of medicinal plants, when we screened out, there are some common secondary metabolites that are commonly present. So some of those secondary metabolites that are common in such anti-helminthic uh, plants, these include glycosides, terpenoids, tannins, flavonoids, alkaloids. When these are found to be common in the anti-helminthic plants that have been used traditionally in our country. So, in an research aspect, if somebody wants to carry out an anti-helminthic activity of a certain plant, then we can screen for such secondary metabolites. Say if the plant is positive with such secondary metabolites, then we can further go ahead with testing of the anti-helminthic activity. So this can be a screening criteria for the anti-helminthic activity medicinal plants. So what is the scientific validation? Like I said, there are many traditionally used medicinal plants that act against helminths. So, in a scientific society, such kind of traditionally used medicinal plants, they need a scientific validation. They need to prove that these such and such plants, they have the property or that can act as an anti-helminthic activity. So once such traditionally used medicinal plants, they are validated, they can be a potential plant that can be a future drugs for such problems in future. So very common foods they're recommended against helminths include papaya, pumpkin, uh, bitter gourd, garlic, pineapple, raw banana, and carrots. These are very common foods recommended against helminths. So, an in vitro experiment of anti-helminthic activity can be done, but prior to that, we need to screen out a plant that can be using uh, traditional therapeutic system or by screening out the phytochemical analysis. So there are different ways to screen out a plant, and after that we can collect the plant, we can make the plant extract by using different extraction methods. Here we are going through Soxhlet apparatus, and after extraction of the plant material, it is being diluted to a certain concentration of our interest. So the plant extract that we make can be diluted in a different concentration depending upon the potential of the plant. So for example here, as an example, it can be 10 milligram per ml, 20 milligram per ml, 30 milligram per ml, or 100 milligram per ml. It can go on depending upon the potential of the plant. So it is diluted using 1% DMSO. So what we can do is we pour the different concentration of the extract in a different petri dishes, and when we place a 4 helminths of choice in the same petri dishes that was made earlier, here the helminth should be of equal sizes of not very aged. So again, There should be a positive control. In positive control, we make use of an albendazole solution. So in negative control, we can make use of 0.9% saline instead of the plant extract. So one part is the plant extract of different concentration, and there is the positive control, which is albendazole solution. Again, in negative control, there is 0.9% saline. So this is how the experiment is set, and after that we need to find the time of paralysis and death for each of the plates. So how do we know that the helminth is dead or it is paralyzed? So when we find that the helminth there is no movement in the helminth, then we can say that it is paralyzed. And when the helminth starts to fade color, then we can conclude that it is dead. So depending on that, the time of paralysis and death is noted down, and we can this is how the experiment can be done. And then a graph can be made for the time of paralysis and the time for that time. So that is how an in vitro experiments can be done. So there are so many traditionally used medicinal plants and against such helminths. So we will find out what are those traditionally used medicinal plants in the next session. So thank you very much. Please like, comment, and share. We need your support. Thank you so much.